Good afternoon, everyone, and welcome to this latest edition of the third Thursday seminar that we're running here at EMB. I'm Paula Kellett, and I'll be hosting today's science session. I'm a science officer at the European Marine Board, and I'm very pleased to see plenty of people already popping in to watch us here. So before we start, just to say, some of you may have already attended some of our events, but for those of you who haven't, this series generally focuses on the science behind our webinars and the science that's being conducted by our members. So next slide, please. Just to give you a few housekeeping rules first. If you do want to ask a question, that's very much welcomed, but we would appreciate if you could clearly add your name and also the organisation and country that you are from, so that we know who is asking the questions. And you can do that by renaming yourself or adding it into your question text. If you have any technical issues, then please use the chat function, and one of my colleagues will be more than happy to help with that. I also note that this webinar is being recorded and live streamed on YouTube. And the recording will be available on YouTube for rewatching at a later date. And we will also be putting copies of the presentations up on our website, so that will be available for you too. So next slide, please. So the focus of this webinar is in fact slightly different from some of the other ones we've had. And this episode will feature our young ambassadors, who will be talking both about their work that they're doing during their PhDs. And they're also going to be talking to us about how they have experienced the Young Ambassador Programme and what that has been like for them. So just before I introduce the speakers, I'd actually like to give you a few bits of information about that Ambassador Programme. So it was launched in 2019, and the format is that we take on 2 young PhDs or postdocs every year from one of our members in our member countries, and they serve a 2-year term with us. So the 2 speakers that we've had today, in fact, joined as the first 2 ambassadors in 2019, and will be rolling off from their terms later this spring. Once they've served their 2-year term, they then join the alumni, or they will become the first alumni in this case. And then they will continue to engage with EMB throughout their careers, hopefully. So the idea of the programme was really to increase awareness of EMB among early career scientists, but also to enable better engagement with early career scientists. Our members are represented on the board by their delegates, and they're usually more senior scientists, so it's great to be able to engage with early career scientists as well. And it's also a way for us to discuss the importance of the marine science policy interface with the younger generation and help explain why it's important to not only do good research, but to present your research and make it available and accessible to policymakers and other stakeholders. It's also a very important way of making sure that the voice of young researchers is heard in marine science at a European level. And our ambassadors then also help us in communicating about EMB, about our activities. In some cases, they can produce communication outputs. Our Instagram account was started by one of the ambassadors. And in fact, last autumn, the ambassadors all wrote blogs about how the COVID-19 pandemic and lockdowns had impacted on their professional and personal lives as well. So some really interesting perspectives and initiatives coming from the ambassadors and really helping EMB in our work. And also they represent EMB at events. And you may have already seen them speaking at some of our own large-scale events, such as the Euro-Ocean Conference held in 2019, and our forum that was held just at the end of last year. So that's kind of giving you an outline about who these people are. So let's move on to the main part of the webinar, and I'd like to introduce our first speaker. Liam Lachs is a PhD student at Newcastle University in the UK, and his science is funded by our member NERC, who is a UK science funder. His work focuses on how climate change affects corals and coral reefs, and it's part of the ERC project Coral Assist. So Liam, if you'd like to share your screen and your video, and I'd like to hand over to you for your presentation.
Thanks very much, Paula, for the introduction. Just let us know, can you see this?
Yep, we can see your screen, thanks.
Great. Yeah, so today I'm going to briefly go through one of the first chapters that I've been working on in my PhD, and also this work was started through an Erasmus traineeship that I did after my master's, which was based in University of Leeds. So it's really been a sort of collaboration between different institutes, this project. And we're looking at population dynamics of corals on Anthropocene coral reefs. For any of you who don't know what the Anthropocene is, that's the modern era that humans are starting to actually leave our own geological layer, our own geological imprint. And you can see that throughout different systems across the planet. Here we're focusing on coral reefs, and coral reefs are hugely important ecosystems for a variety of reasons. What you're looking at here is a satellite image of the Great Barrier Reef, highlighting one of the major functions of coral reef ecosystems in terms of what humans get from them, which is protection from storms. Coral reefs form huge barrier systems which protect coastlines. They're carbonate producing organisms. They're also producing a lot of the sand which lines the beaches in these areas. They're beautiful ecosystems and completely heterogeneous. You can see here these outlines of coral reef edges with these sandy lagoons, and this is really important for human livelihoods as well. There's hundreds of millions of people throughout the world whose livelihood relies on fisheries or tourism, which is relating to work on coral reefs. But they're not just important for humans, they're also important for biodiversity itself. It's estimated that a quarter of the biodiversity in our oceans is actually found in coral reef habitats. But coral reefs, they're actually made from these reef-forming corals. And here you can see a reef flap with a small drop-off going to a deeper area. And all of these coral individuals, they're different species. So we're starting to get a picture of how diverse, how complex these systems are. And each of these species has its own life history strategy. It lives within a population, which has to have birth rates, death rates, growth rates. So they're all viable populations. And that's the focus I'm going to take here, thinking about coral reefs as assemblages of coral populations. Coral reefs are really threatened from climate change. Here you can see projected sea surface temperatures for Palau in the West Pacific over the next century. And even under the best case scenario, we're committed to a certain degree of warming of our oceans. If we continue in our carbon emissions as business as usual, this degree of warming could be a lot higher. And that has serious implications to corals, because corals bleach and can suffer huge mortality due to heat waves and heat stress. What is coral bleaching? I'm sure you've all heard of coral bleaching, but what actually is it? Well, corals are animals, they're not plants, but they do have a symbiotic relationship with algae cells. And here you can see the coral host with its tentacles. It's closely related, it's a cnidaria, so it's related to jellyfish. And in here, photosynthetic algal cells. Well, when the coral suffers from heat stress, this symbiosis breaks down and those algal cells are expelled from the coral host, leading to starvation and potentially mortality within weeks. This is happening on mass scales and is a really pertinent issue under climate change. But monitoring coral reefs is often there's often a focus on getting out in the water, diving, and recording the percentage coverage of different types of coral. But what I want to think about here is populations. And a really simple sort of idea of a population is this predator-prey dynamic of a fox and a hare. Obviously, there's mortality on the hare population due to the fox predation. There's also going to be growth rates, which helps the population boost back up after a disturbance, growth of those individuals, and again, mortality. So this is really how we have to think about populations, as interbreeding within a species. Now, if we move this on to coral reefs, consider these two hypothetical reefs. On the left side, you've got a lot of small-sized corals, and the reef on the right side, you've got a few very large corals. In this case, if we look at the percentage cover of live corals of this reef, it's much higher for reef B than reef A. But if we just push this through a very basic kind of thought experiment model, where every time step from each reef, one coral dies, then quite soon, reef B has 0% cover left. And thinking about these populations of corals as populations can really help us to understand the resilience of these systems. Now, linking this onto some actual real data. So this is a study that's linking size structure, heat stress, and coral bleaching in the subtropics. And it's, well, what you're looking at here is the species we're interested in, Pocillopora elysei, and it's undergone a mass coral bleaching episode only a few years ago. This species has a life history like any other organism. Here you can see some of the recruits. They grow, and these adults are providing important habitat for some species of fish here. And this highlights the sort of overall growth form of this coral. Now, this study was focused on 4 islands in the Solitary Islands Marine Park, which is in Australia. They're sort of in mid-shelf and offshore islands with slightly different hydrodynamics. It's warmer offshore, actually, in this case, due to a current that comes from the Great Barrier Reef pushing warm water south. And from the effort of dozens of really devoted scientists, there's a 20-year time series of information from these islands. So there's been surveys of the corals over the last decade, and we also have temperature data from in situ temperature loggers that are on the seabed at these islands over the last 2 decades. So linking that together, we're going to try and get a bit of insight into the population dynamics here. So what do we do in these demographic surveys? Basically, using scuba, dive down on these reefs, run transects along the seabed, taking photos of the seafloor, something like this. You can see here the magic wand is the scale bar for this photo. Then in post-processing, get all of these images and circle all of the corals of interest. And that gives you the size of these corals. When you start to put that all together, you get an idea of the size structure of the population. Now, in 2016, there was a huge marine heat wave in the Great Barrier Reef, Eastern Australia, the Solitary Islands, in the beginning of the year. You can see here the red line is the 2016 in situ temperatures, and it's almost 2 to 3 degrees higher than the baseline average temperature. We use this stress threshold in blue to define a metric which is called degree heating weeks, essentially a metric of heat stress, which says that within 2016, the maximum heat stress was, I think it was a value of about 10. And this led to enormous bleaching throughout the coral community, throughout the Pocillopora population, should I say. In this case, you're looking at a healthy coral moving to a half-bleached coral, and 6 months later, there was a lot of these dead corals covered in algae, which has a severe implication on a population when you lose almost all of the adults. So if we actually want to have a think about this time series, how did these population processes play out over time? Well, back in 2010, there was a really interesting shape of the size structure. In this case, the dotted line is the sort of line between juveniles and adults. In 2010, there was a huge peak in the numbers of juveniles and really small corals we were seeing, highlighting there was some sort of recruitment pulse in this year. 2 years later, you can see that the peak of that distribution has shifted up. That represents some growth of this cohort and continued growth up until 2016. But the bleaching episode in 2016 caused drastic reductions across all of the different size classes, and it hasn't recovered yet. Critically, this recruitment pulse hasn't been seen again, which is a bit of a worry. Now, if we try and think about these population dynamics, how do these actually link with heat stress? So we're going to first of all look at the juveniles and then the adults, and here for each year of survey data, we pair it to the maximum heat stress in that year. It's really clear here for the offshore sites, there's only recruitment pulses happening in those low heat stress years. It seems like in the high heat stress years, there's some suppression of recruitment, whereas for the adults, we haven't seen this type of trend, meaning higher sensitivity of those earlier stages to heat stress. So to wrap things up here, coral bleaching caused huge declines across all the sizes of this coral population, and recruitment seemed to be dependent on low levels of heat stress or a lack of heat waves. This really brings into question the viability of these populations under climate change. If marine heat waves become more and more frequent, if we start to see less and less of these big recruitment events, can these populations persist? The benefits of this approach that we used was by looking at the size of corals, we're really able to monitor population processes, get an idea of recruitment and mortality. It gives you a lot more understanding of the population than just looking at the percentage cover of corals. But one of the drawbacks of this was the amount of time it took to analyze all of these photos. We're talking about in excess of 2,000 photos and measuring the sizes of in excess of 6,000 corals, I think. I'll have to double check those numbers. But it's a huge amount of manual effort to do this work. Where do I see this heading in the future? Clearly, the insight that we can get into coral populations, meaning the insight into coral reefs, can be really quite, you can get a lot of insight from using this kind of approach. But there's also huge advancements coming in the technology that we're able to use throughout social media and Google. All of these different systems are using machine learning to classify images. So I really see the future of this field is linking these kind of in-depth demographic surveys with machine learning algorithms which can classify those images automatically, circling those corals automatically. And that would lead to really improved understanding of these systems, potentially at a near real time. If you can do surveys, take pictures, plug it into a machine learning system, and then get an immediate inference about the population. And obviously understanding these systems better is going to help in improving our management and governance of these ecosystems, but also improve how we understand their future and how we plan policies for climate change and different things. So this is the overall discussion of my first chapter. And yeah, I have to mention again, this has been a huge effort from numerous different groups. So thanks so much to everybody who's been involved in this. So I'd now like to change a little bit, just to have a quick mention of the work that I've been doing as a young ambassador of the European Marine Board. Essentially, my involvement at the beginning was to come to EMB and learn how the organization works, see how they fit into the landscape of policymakers, scientists, and bridging that gap in between. I then attended a lot of different events, from British Ecological Society meetings to Euroceans, had the chance to talk at UNESCO and basically promoting the sort of integration of science into policy at these different events. And my current work, continuing with European Marine Board, is with their new working group on coastal resilience. We've been meeting over the last month or two, and the aim of this is to come up with a document which really outlines some of the important points that we need to be looking at to improve coastal resilience in European coastlines in the coming years. So yeah, thanks everyone for listening, and I welcome any questions.
Thanks very much for that. That was really interesting, and I certainly learned something. I'm sure everyone else did too. So I've seen there's a couple of questions specifically relating to your research, so I will ask those now. And if there are questions relating to the ambassador program, then I think we'll save those to the Q&A after the second speaker. So I've seen two questions about your work. The first one is regarding the recruitment event that you mentioned in 2010. Yeah. And you said you haven't seen another one of those. I mean, do you have a feel for how often those tend to occur, or how often you could expect those to occur in what could be considered normal situations?
These recruitment events, I mean, what we saw here, I'll go back to the time series. I've really just showed data from one site throughout this time series. And we saw similar things in the other offshore site. There's an assumption about some tropical coral reefs that recruitment can be relatively constant, and it doesn't seem like that's what we've seen here, because there was this huge amount of recruitment, and there's a clear cohort growth of those recruits throughout the coming years. The question of what was happening before 2010 is really good, because we don't have data from them. I mean, was this population having a huge recruitment pulse like this every year before 2010? It's possible, but I feel that if that was the case, then there might be a different shape of the distribution even in 2010, perhaps more of a rounded distribution. These events are also like super stochastic. They're really random. It depends on the weather. If you've got howling winds when the corals spawn, all of that spawn is going to drift off. If it drifts out into the open ocean, it's no good for anyone. So it's really kind of a bit of a sort of luck process as well. But for these kind of systems that seem to be reliant on these sporadic events, if they're threatened by heat stress, that's a bit of a worry, really.
Yeah, indeed. So there's another question from Virgilio Ratunil in the Philippines. And the question was, how did you select the sampling sites for the coral population dynamics?
I'll go back to my map. So this, I mean, I haven't been working on this since 2010. It was Bridget Summer and Maria Bager and John Pandolfi's lab in Australia who started these surveys back then. And actually, you can't see it here, but this is all part of a much bigger kind of study area, where basically there's sites going all the way up to the Great Barrier Reef and even going south beyond here, really looking at these latitudinal gradients. And there's actually a PhD student in Hull, Fiona Chong, who's actually looking at these kind of analyses for the entire latitudinal gradient, which is bound to produce some really interesting results in the coming years. The reason why for this study we chose these 4 sites is because of the fact that there was these temperature loggers at these sites as well, because not all of the sites would have such a sort of regular monitoring protocol in terms of temperature.
Yeah, indeed. Yes, you need all the combination of information before you can make a full study. There was another question from Rupert Simon, or Simon, sorry, I'm not sure of the pronunciation, asking, have you considered using aerial monitoring to calibrate your underwater research? Apparently Belgian scientists have been using sort of underwater photography to calibrate satellite images and to monitor developments. Is that something you considered?
I mean, I'm actually currently working on a data set of coral bleaching, which is, some of it comes from aerial surveys. But with that kind of approach, you're not going to get the same resolution as getting into the water. I mean, especially sometimes in these temperate seas, the water can be quite murky. Maybe you don't really see the bottom from outside. So to get this really high resolution demographic understanding, getting it from aerial surveys or satellites is going to be really tough. But then you have things like the Catlin Sea View survey, which, for people who haven't seen that project, it's really interesting. It's worth checking out. And it's these big scooters that pull you underwater as a diver with really, really fantastic cameras. And they can cover kilometers of reef because they're not relying on their leg power to swim. And I guess linking in with those kind of technologies to improve the spatial extent of the surveys and things, which is probably more feasible for demographic kind of assessments.
That's great, thanks. And there's just one more question before we move on. So again, from Virgilio in the Philippines, and he was asking, what are the appropriate management for bleaching corals?
This is a really good question, because if we're talking about mass coral bleaching, which is an effect of marine heat waves, what's a management strategy? You can't simply say, oh, we predict a marine heat wave is going to come. There's nothing you can do about that. The one thing that we can do is curb our carbon emissions, and that's, at the end of the day, that's the underlying thing that needs to be addressed to avoid these climate impact issues. But there's other things people are looking into. For instance, the Corallisis Lab is looking into selective breeding corals for heat tolerance, trying to understand if can you implement restoration of coral populations and coral reefs to support some of the key ecosystem services that they provide. You won't be able to restore systems to a sort of historical level, but maybe some small areas can be kept at a reasonable level that at least fulfills some of the services that we get from coral reefs.
That's great. Thank you very much, Liam.
Thanks.
I will now share my screen to introduce our second speaker. So our second speaker will be Alba González Vega. She is a PhD student at the University of Las Palmas de Gran Canaria, and also at IEO in Spain, and IEO is one of our member organizations. She's also been a Young Ambassador since 2019, and her work focuses on the submarine volcano Tagoro, and she's going to tell us much more about that study in her presentation. So I will again stop sharing. Alba, if you could share your screen and turn your video on, and the floor is yours.
Thank you very much, Paula, and thank you all for being here. I'm going to share my screen. Okay. 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 So thank you all for being here. My presentation will have two parts. Mostly I will talk about my PhD research, but at the end I will also take a few minutes to talk about my experience as a young ambassador for the European Marine Board. So, sorry. My PhD research is about a submarine volcano, which is called Tagoro, which erupted in 2011, so 10 years ago already, at the south of El Hierro island in the Canary Islands. And as you can see in the pictures, it was very close to the coast, very close to a village called La Restinga. So here's a bathymetry of the volcano and its location. So you can see it's almost 2 kilometers away from the southern coast of the island of El Hierro. It has a diameter of 1 to 1.3 kilometers, with the summit at 88 meters depth, and the main crater, where most of the emissions come from, is at 127 meters depth. So it's relatively shallow in comparison to the submarine volcanoes that are typically studied, which are very deep. So the eruption of this volcano, like I said, it erupted in 2011 in October, and the eruption lasted around 6 months until March 2012. And during this time, as you can see in this satellite image, the emissions affected a very wide area around the island, and there were obviously a lot of physical and chemical changes in the water column. So for example, the temperature obviously rised. The maximum was about 18 degrees above the normal conditions, and the salinity decreased. There was also a lot of CO2 emissions, which caused a very big decrease in the pH, and the minimum pH that was reached was of 5 units. And there was also a lot of emissions of reduced iron and reduced species of sulfur. And so all these reduced compounds had to be oxidized when they come in contact with seawater. So this caused a very big depletion in oxygen. And it even reached anoxic levels in the area at some point. So that was the first 6 months. And then after that, the eruption ended and the volcano entered a new stage called the post-eruptive stage, or the degassing stage, which still remains present. And in this stage, all this wide area that was affected at first is no longer affected, but the emissions stay in a much smaller radio around the main cone of the volcano, about 500 meters around the main crater, and very close to the seafloor. But still in this small area, we find all these anomalies that I talked about. And the emissions are mainly through hydrothermal vents and chimneys, and also by diffuse flow from the floor. So here's a timeline so that you can have an idea of how much we have sampled this area. So as you can see here, each of these little arrows is one oceanographic cruise that we have carried out, where we have taken samples and data for physical, chemical, biological, and geological studies. So it has been very intensely monitored. And also we have used different kinds of methodologies for the sampling. So at first we used the classical oceanographic methodology, which is vertical profiles with a rosette, with a CTD and with Niskin bottles. So just going down and up. But then, especially in the post-eruptive stage, as we saw that the emissions stayed very close to the seafloor, we used a more specific methodology, which is called tow-yaw, because you are towing the rosette through the seafloor and going up and down at a very low speed. So you have all the area close to the seafloor, you have it covered. And then also in some of the cruises, we have been able to use an ROV, and we put a syringe in it so that we can take samples directly from the hydrothermal vents. So my PhD research is based on 3 main areas regarding this volcano. So first, we wanted to study the oxygen depletion that happened during the eruptive stage, like I said, because of all the reduced compounds that were emitted. Then we also wanted to study the emission of inorganic nutrients from the volcano, both in the eruptive and post-eruptive stage. And this study is actually already published, so I put the reference here in case you are curious and you want to read it. And then the last study, which we are still working on it, is about whether these emissions of nutrients have had a positive effect on the local ecosystem. So I will talk a little bit about each of those areas. First of all, like I said, we studied the deoxygenation or the oxygen depletion that happened during the eruptive stage. So for example, here in this profile, you can see in black a reference profile. So this is the normal conditions of oxygen in the water column in that area, which, as you can see, is around 200 micromoles per kilogram. And then in red are some of the most extreme profiles that we measured during the eruption. And you can see that the minimum was of around 7 micromoles per kilogram, which represented a 96% decrease in oxygen in comparison to normal conditions. So obviously this caused a lot of mortality in the area for fish and zooplankton and so on. Here's so that you can see the spatial distribution of this deoxygenated plume. Typically, it had this structure that you see in figure A, where it was, because of the local current, it's usually transported southwest, and it had an area of about 378 square kilometers. But then also at some specific times, when there were, for example, changes in the local currents, it could reach even farther regions, like we see, for example, here in figure B, where it reached the north of the island. And we even saw at one point that the volcanic plume was transported by an anticyclonic eddy as far as 80 kilometers away from the volcano. You can see it in the satellite image, and here you have the oxygen profile at that point, where we still had an 8% decrease of oxygen, which is a lot considering that it had been transported for 80 kilometers. So the second part of my research was based on the emission of inorganic nutrients from the volcano. So we studied 3 nutrients. One is nitrate, which is actually nitrate plus nitrite, but we studied them jointly because there's not a lot of nitrite. So then phosphate and silicate. And as you can see in these profiles, the black profiles are always a reference profile, so the normal conditions. And then we have in orange red, we have the data from the eruptive stage in the whole water column. And so as you can see, it was a lot of nutrient emissions, but it didn't matter because the ecosystem was negatively affected by the oxygen and pH and all these things. And then the rest of the data are from the post-eruptive stage, so the one that we are at right now. In blue, we have data in the water column, and then we have the data with tow yaws near the seafloor in purple, and then some data from taken with ROV from the vents in green. And something, well, as you can see, all the nutrients were very largely enriched. But something interesting is that if you look at the different kinds of sampling methodologies, you can see that for phosphate and silicate, they were more concentrated the closer we were to the source. So in the vents, they were the most concentrated, and near the seafloor, it was a little bit less, but still a lot. And in the water column, it was not so much. But in nitrate, we don't see that that behavior. We actually see that there's more in the water column than in the vents, for example. And we later discovered that this is because nitrogen is actually emitted in the form of ammonium, and then it undergoes some processes and it's converted into nitrate, and that's why there's more nitrate in the water column. But we did, when we started measuring ammonium, we did find also very large enrichments in the vents. Here is also the spatial distribution of the nutrient plume. In this case, it's a vertical contour, a vertical section with the bathymetry of the volcano. And the emissions reach about a 400 meters diameter around the main cone. And something interesting is that, well, the emissions usually don't reach the surface, but depending on the time of the year, they can reach the mixed layer and the euphotic zone. So obviously, when we saw that, we started wondering if these emissions are fertilizing the area and are feeding the phytoplankton or the primary producers, and are having like a positive effect on the ecosystem. And that's the next step that we have to take. We still don't have, this part is still undergoing, so we only have some preliminary results. Like, for example, here you can see that we found some positive anomalies of oxygen and fluorescence very close to to the volcano in comparison to the normal amounts that we usually find. So this can indicate a higher primary production, but obviously we still need to carry out more studies. This is only preliminary. What we do know is that, well, like I said, in 2011, the whole local ecosystem was very drastically and negatively impacted. And now in the area, there is a lot of life. The ecosystem is completely restored. Some say that there's even more life than before. And what we want to find out is if the ecosystem sort of recovered itself, or if the volcano actually helped with that recovery. So that's all for my research, and now I want to take a few minutes to talk about my experience as a young ambassador for EMB, because my time as an ambassador is coming to an end. So I want to take this opportunity to sort of reflect on this time and what I've done and what I've learned and so on. So the ambassadorship lasted 2 years, and as you can imagine, the first year was very different from the second year. So 2019 versus 2020. At first, there was a lot of travel and in-person events, and the second year in 2020 was mostly Zoom calls, etc. But that didn't stop us from getting involved and doing a lot of activities. So we just had to adapt a little bit. Here is a very general summary of the activities that I have carried out. Some of them were already mentioned by Liam and by Paula. So mostly, well, first we went to visit the EMB headquarters in Austin. We attended many events by EMB, like plenary meetings, the Eurasian Conference, the 7th EMB Forum. I also talked about EMB in other places, like in conferences within my own networks here at EAO and at the university. And then also, like Liam said, we have been, well, each of us has been involved in one of the EMB working groups. I've been involved in the geohazards working group pretty much from the beginning. And it's been really interesting. And then we have also done other small contributions like blog entries, the Instagram account, etc. And from this experience, I've had a lot of benefits. Obviously, I've learned a lot about marine science policy in Europe. I've also broadened my networks. I've met a lot of different scientists from very different fields, which has been really interesting. I've also learned a bit about how international collaborations are made. Like, for example, especially through my involvement in a working group, I've seen how it is to create like a document in collaboration with lots of different scientists from different institutions from different countries. And mostly, I think it's made me think about the way we communicate our science, because I think as researchers, we tend to be like very trapped in our own bubble. Like we publish our results in very specific journals, and those papers are read by other scientists from our area of expertise, and it's very closed up. And I think it's important to remember that all that knowledge has to be shared with a broader audience, and also with politicians when needed. So I think that's important. And additionally, it's also helped me with some personal skills like communication, initiative, confidence, etc. So that's also, I just want to thank one more time the European Marine Board for this opportunity. And I want to encourage all of you to support these kinds of opportunities for early career scientists, because I think they can be very beneficial. So that's all I had to say. Thank you so much for listening.
Thank you very much, Alba. I mean, lovely words to end on there. We certainly can't complain about that. So I've seen there's a couple of questions come up about your research specifically. So I'll start with Virgilio, again from Philippines. He's very engaged, and that's great to see. He asks, has the eruption event attracted some marine organisms that are not necessarily endemic to the area during this post-eruption stage?
Yes, I mean, the communities have definitely changed. There are some published studies about like the benthic communities that are living there, and obviously there was new substrate formed, so there were new colonizers in that area. And also there was a new species of a bacteria that was newly discovered in the area. So yeah, there's been definitely a change in the structure of the community at all levels. Yes.
That's really interesting. Yeah. And my question kind of follows on from that. I mean, you've been studying this volcano for 10 years. Are there plans to continue doing that and continuing to see how it evolves?
Yeah, definitely. We actually have like a structural plan within the IEO to sample. So we have 2 oceanographic cruises per year. And so far it seems like this is going to keep going, because we have one of the biggest time series of data from an active submarine volcano. So that's, we can't lose that. So for now, I think, and especially because we haven't stopped producing new scientific knowledge from this. So every year we publish new papers on new topics related to the volcano. So yeah, we're definitely going to keep doing that.
That's great to hear. We'll look forward to what keeps coming out of that. There's also a question from Sheila. She says, do you know if there have been previous eruptions in the area?
I mean, the whole Canary archipelago has a volcanic origin. So there's always, I don't know if specifically in that area, I mean, probably at some point, like I said, everything was originated from volcanoes. But the interesting thing is that this volcano is actually the first submarine eruption that was recorded in the islands from like 500 years that we have of records. So like from, since the islands have been, people have been living here that can like record what happens. This is the first time that a submarine eruption has been recorded. But we have like, I would say approximately 1 to 2 eruptions per century. So for example, there was most of them are like subaerial eruptions, so on land. We had one in the 70s, like in 1971, I think it was, and another one at the beginning of the 20th century. So there's a lot, but specifically in that area, I'm not sure when was the last one, but there was definitely a lot of them.
Yeah. Well, as you say, it's quite an active area in its own right. There's also a question from Rupert Simon in Finland. He says, you mentioned iron and manganese concentrates, but you didn't present the data on that. Could you elaborate any more on what happened with the iron and manganese?
I don't think I mentioned manganese, but I did mention iron. Yes, I didn't present any data because it's not my research. This has already been published. But yes, we have measured a lot of we have made a lot of measurements on dissolved iron, and there have been enrichments of, I think it was like up to 5,000 times respect to normal conditions or something like this. This is published, and it's really interesting. So yeah, because it's also, I mean, I have studied these nutrients that I talked about, but people say, okay, but what about iron, which is also a very important nutrient for phytoplankton and all this. But yes, iron has also been is also being emitted in large amounts, and it can stay, because iron is usually quickly oxidized, but in this case it can stay for a fair amount of time in the reduced form.
Yeah. And I think there's one more question from Sheila here. So do we know what the long-term impacts from eruptions were, and if the extra nutrients have had a positive impact on the ecosystem?
Yeah, so this is basically the third part of my PhD. We're working on it. I mean, we think that the nutrient emissions have had a positive impact, or are having a positive impact. But yeah, we're working on it. We have taken some samples of, we have done studies of primary production in the area. We have also taken samples for to study the abundance of phytoplankton, bacteria, and all this, and to see also the structure of their communities. So we have a lot of data that we've collected. We are also collecting right now. We have a cruise next month, but this is still ongoing, so I can't give any definite answers, but we think that yes, there is a positive impact on the communities.
That's great. Perhaps if Liam could turn his video on as well, and then I can see if there's any general questions sort of about the ambassadorship and about about your work in general. So, I mean, a question to both of you. You wrote some great blogs about the impacts of COVID on your research, but instead of sort of looking at the negatives, what positives can you take from what the pandemic has done to your research? Perhaps Liam, if you go first.
Yeah, well, since the last year, well, yeah, it was this time last year, we were planning with the Coral Assist Lab in Newcastle. We were planning to do our field work in Palau, had flights booked and everything. One of the PhD students got out there, and she's still there, and the rest of us are still in the UK. So it was a real sea change in terms of, talking about, there was some time we were talking about just delaying, but at some stage you think, right, we need to reevaluate what our goals are here. And we were having these discussions about coral bleaching and the different drivers of coral bleaching. And I started to work on some remote sense data from satellites, linking temperatures and coral bleaching data. And I mean, I learned some really valuable things, a lot of stuff about statistics that I wouldn't know otherwise, using the high performance computer facilities in uni, which gives you a lot of computing power to do some quite advanced stuff, which has also really been really sort of fun and is probably going to stand to me well. And it also led to a collaboration then for that chapter with NOAA, the US National Oceanic Atmospheric Administration. And I'm not sure if that collaboration would have been as easy if everybody, and we're also accustomed to just Zooming for everything now. I think people are really open to collaborating in this sort of time as well. So there's definitely been a lot of positives to pull out of it. Saying that, I'm really looking forward to getting back to actually diving on a coral reef and getting my hands dirty again with that stuff. So yeah.
Yeah,
I can absolutely understand that. And Alba?
Yeah, so I think, for example, one of the impacts that COVID had was that we had to cancel some of our oceanographic cruises, which is a negative, but also that kind of forced us to work with the data that we already had, because because we are always collecting so much data and we have such big time series, sometimes it's we don't have the time to do something with all of it, and some of it just kind of stays there. So this forced us to look at what we had and how we could work with that. And so I think that was a positive. And then I also think, and this is probably a shared opinion with many of you, I also think that COVID helped us see which events need to be in person and which events can be just a Zoom call, and you don't need to go very far away to have a 1 hour meeting or so I think that's also positive that we have gotten used to this kind of communication, even though I'm really looking forward to when we don't need to do everything online. But I'm also glad that now we're more comfortable with using the online tools that we have. That's my opinion.
Yeah, well, I think this is a very good example indeed. There's a question from one of our other ambassadors, so that's great. They want to know what's going on too. So Alessandro asks, did the ambassadorship stimulate you in improving and communicating your science to people from outside your field of expertise? So again, Liam, I'll go to you first for that one.
I mean, definitely is the answer, because also it depends on what your sort of past has been, but like if you've gone through the academic system, degree, masters, doing a PhD now, and I've done other internships in institutes and stuff as well, but overall you're always in the academic sphere, and there's a certain way of thinking about things, there's a certain approach that maybe just isn't as kind of broadly available. So going to events where you're talking to people who are maybe in the higher level management of some marine research institute, or maybe they're actually working in European Commission, these kind of things, and you realize, okay, their priorities are different, and it's actually, you have to think about things from a different perspective. Yeah, for sure, doing this ambassadorship has helped me think about that stuff.
Yeah, that's great. And Alba?
Yes, definitely. Like I said, this is a good way to like get out of our bubble and look at other areas of expertise, and also kind of think about how to communicate science for people that are outside of this bubble. And so yeah, I think it's been really, it's helped me a lot with thinking about the best way to communicate our science, and also adapting it to the public that is going to hear it or read it or whatever. So yeah, definitely it's helped a lot.
Yeah, that's great. I'm just going to ask one wrap-up question, and I think we're running out of time, but I mean, do either of you have a specific highlight from the 2 years that you'd like to share? I'll go with Liam first again. Sorry to put you on the spot.
I don't know. I'm just trying to think.
Alba, if you have one. Oh, sorry.
One of the funny I don't know if it's a highlight. One of the funny situations was back when we could actually travel, and we'd come to the European Marine Board to actually start. We did this sort of 2-day course to understand how European Marine Board fits into the science policy landscape. I think it was that visit where I ended up getting stuck for an extra amount of time because there was a huge storm coming in, and the flight that I was on was this tiny plane, which I didn't want to be in that on during a big storm. That was a funny situation. Not exactly a highlight.
Maybe a low light. Alba?
I don't know. I think maybe as a highlight, I always remember when we gave a little speech at the end of the Eurasian conference, because it was also like very impacting, because we were in the UNESCO headquarters in Paris, and it was like I was like, oh my God, I can't believe I'm here. And then we got to speak for a few minutes, and I think for me that was really, that really stuck with me. It was really cool.
Yeah, I second that.
Yeah, I think we all do. I mean, that was definitely a great way to make your entrances as our ambassadors, I think.
Yeah.
So thank you very much, both of you. I'll just share my final slide with everybody. There we go. So yes, thank you very much, first of all, to our speakers. It's been really great to hear from both of you all about your research and about your experiences. Thanks to everyone else who's engaged in this webinar. It's been great to have you here and asking questions and getting involved. As I said, the webinar has been recorded and will be available on our YouTube to rewatch. And the presentations will also be made available on our website. So if you keep an eye on that, you will be able to find the information there shortly. This is a monthly webinar series. So the next webinar will take place next month on Thursday, the 15th of April at the same time. And that will be on marine citizen science, trends and frontier issues. And it will specifically highlight some of the latest activities that have been going on in the UK and Ireland in this area from 3 great speakers. So if you're available, then do sign up for that. The registration is open at the website that you can see here. And we'll be adding information on upcoming editions to that website regularly. So, do make sure you keep track of what's going on. that's all from us now. I'd just again like to say thank you to all our speakers. Thank you to everyone for being here, and have a great afternoon. Goodbye.
Goodbye. Thank you, everyone.